Hello, my name is Lluis Montoliu. I'm a researcher at the National Center for Biotechnology in Madrid, Spain, and I will be presenting in a few slides what I think the animal models have been contributing to the field of albinism. I'm going to be sharing my presentation right now. So, animal models has been a very active field in order to advance our understanding and our proposals for treating different forms of albinism. So what follows is my take, my view on how this field has been moving along on the past years. And this has been prepared particularly for the International Scientific Conference on Albinism 2022, ISCA2022, that is being organized by the Global Albinism Alliance. So albinism is a genetic condition, is a recessive trait that affects between 1 to 10 to 1 to 20,000 people in the world with different proportions depending on the countries. And it is normally characterized by people that are visually impaired because they have a number of retinal alterations, such as fovea hypoplasia. Most people would lack fovea, which will result in a reduced visual acuity, but also they will have abnormal chiasmatic connections, which also is related with nystagmus and with photophobia. There are several types of albinism. There are some are syndromic, which means that are affecting different cell types in the body beyond the pigmented cells in the skin, the hair, and the eyes. And also we have oculocutaneous and ocular albinism. So genetically, this is a diverse genetic condition because we know at least 22 genes whose mutations are associated with different types of albinism. So as I said, one of the main features of albinism is that people with albinism have this foveal hypoplasia. So the fovea is the central region of the retina where we have the highest concentration of photoreceptors. We have the highest visual acuity. The rest of the retina is considered to be the peripheral retina and it's normally processing a low resolution image. As you can see in this eye fundus above, we see a person with albinism without a fovea, and below we see a pigmented retina with a macula that is pointing us where the fovea will be. So beyond this hypoplasia, people with albinism, they have abnormal connections between the ganglion cells of the retina and the different visual nuclei in the brain and the central nervous system. And this is resulting in altered 3D vision. So the stereoscopic vision is altered in these people because they mostly lack what we call the ipsilateral projections of these ganglion cells. So basically, the first thing to mention and to summarize here is that albinism, in spite of what is been historically been considered to, normally years, this was considered a dermatological condition, albinism that is associated with hypopigmentation in some cases, not in all cases, mainly features a visual deficiency. And this is what we have tried to reproduce in mice, also the alteration in the pigmentation. So all what we see regarding this visual deficiency is established during embryo development. So here we have on your left we see an albino retina, and on your right, you see a pigmented retina. These are mouse retinas. And as you can see, basically they are essentially apparently the same, with the exception that the retinal pigment epithelium and the melanocytes colonizing the choroid are pigmented, obviously, in the wild-type retina, whereas they are albino, so you don't see melanin, you don't see the pigment in the albino retina. So we know that due to this lack of melanin, directly or indirectly, there is a distortion in the program, in the developmental program, that results in these retinal alterations that eventually they develop into this visual deficiency. Genetically, as I said, we have 22 genes, and mutations in these 22 genes are associated with albinism. Here we have listed the 22 mouse genes, and in the above, highlighted in yellow, are the non-syndromic type of albinism. Below, in pink, are the syndromic types. There are two types of syndromic, Chiari-Akihashi syndrome and Hermansli-Putlag. We have one type of Chiari-Akihashi, but we have 11 subtypes of Hermansli-Putlag. And these can be more severe forms of albinism. Whereas the non-syndromic parts, we have 8 types of what we call oculocutaneous albinism, from 1 to 8. We have ocular albinism type 1, and we have some new type of albinism that is called fonda, which is a complex phenotype as well. So how do we investigate albinism in people? Obviously, we cannot use people. We need to use another in vivo approach. Ethically, it will be unacceptable to use people with albinism to perform investigations. So we need to go as close as possible to the human beings. And of course, the closest situation would be to choose an animal model which is related to us. And of course, this could be perhaps a primate. You might remember this is a gorilla, the only known albino gorilla in the world, was living for years in the zoo of Barcelona. It was called Snowflake. Eventually, in 2003, he was euthanized because he was suffering from a very severe skin carcinoma that had already metastasized, and he had to be euthanized. Of course, there was only one, so there is not much we can see, and there is not much we can do. Plus, as you know, perhaps in Europe, we are not supposed to be using these big primates. We are not supposed to be using gorillas or chimps or bonobos or orangutans in our research. These are essentially prohibited. And this means that we need to find out other type of animal models to do our analysis. So the most popular animal model that we have found out over the years are mice. So let's see how studying different types of mice has been successful and has been positive and instrumental in our understanding of albinism. So essentially, with mice, because we have many different types of mice, many different genetic strains, and because we can virtually reproduce almost all types of mutations, we can mimic what we see in the human population. And actually, we can prepare and we can investigate mouse models of albinism. And this is something we can do because even though externally we might look very different, when you would be looking at the embryo development, a mouse embryo and a human embryo at the early phases of development, they are very, very similar. Later on, they will diverge and they will eventually result in animals that are very different. But at the very beginning, we follow the same path. And this is because we share most of the genes. The best way of illustrating this is this graphic in which you see at the bottom, you see these squares with 23 color squares representing the 22 autosomal chromosomes and the sex chromosome, in this case, the X chromosome. And above, what you see are the 20 chromosomes that are normally present in the mouse cells. And you will see that every single human chromosome has been represented somewhere in the mouse genome. Sometimes we have syntenic regions of chromosomes, which means there are regions that have been traveling together. Sometimes there have been some cutting and pasting and some reorganization, and this is why we see a bit of mixture. This is during the evolution. But essentially, the message to be inferring from this slide is that we can expect finding nearly all genes that we have in the human genome in the mouse genome. This is to say that we can generate a variety of mutations, and we can generate many different mouse models, almost representing mutations in every single gene of the mouse genome that is homologous, that is orthologous to the corresponding gene in the human genome. For instance, I told you that in general, albino mammals have this foveal hypoplasia, which is most severe in primates, where in people, in human beings, they usually lack fovea. You've seen these pictures before. You've seen these fundus of the eye, and you see on the left, you see an albino retina, whereas there is no fovea, and on the right, you see the pigmented retina with the fovea. But we have a problem with mice, because unless what happens in primates, we have a very peculiar distribution of photoreceptors in primates. You see that the cones, they peak around this fovea, and that's where almost no rods are present, but most of the cones of the retina are peaking in this area. And this is what happens in human beings, whereas in mice, what we have is a photoreceptor distribution that is rather different. We have a central retina, which appears to have more photoreceptors, but there is a very weak decline towards the peripheria. So there is no much difference. So in other words, mice do not have a clear fovea, but mice have a central retina that has more photoreceptors. How can we address this? How can we investigate this? Well, what we can do is we can count photoreceptors, and we know that at the central retina, albino mouse retinas have a deficit, have around 25–30% less photoreceptors as compared to the wild type. And of course, when we will restore the expression of a gene that has been mutated, such as tyrosinase, then you can restore the pigmentation, and you can restore as well the number of photoreceptors. So even though mice do not have fovea, we can still ask the mice whether the central retina has recovered the proper number of photoreceptors as it should be in the case of the wild type. There is another animal model that we have used in the past. These are the rabbits. Rabbits are not rodents. Rabbits are lagomorphs. And rabbits do not have a spot, do not have a fovea, rather they have a line. It's like you would take a fovea and you will extend the fovea, and this is called the visual streak. So basically, they have a very long fovea that is crossing across the retina. And again, albino rabbits, they have a decrease in the number of cells that are populating this albino, this visual streak, as compared to pigmented rabbits that they have a normal number of cells. This is something we investigated some years ago. And in principle, even though for fovea it would be even more instrumental to use rabbits. Rabbits, they represent another additional complexity, and they are not that amenable for genetic manipulation as we can do now with mice. With mice, we can also investigate these abnormal visual pathways because we can do this anterograding, we can do this labeling, we can add a neuronal tracer in the optical cap, and we can investigate how this neuronal tracer is permeating and it's traveling towards the brain, towards the nuclei. And if we do this, we will see that the pigmented mice, if you see the bottom slides, they will distribute some fibers. They will be not only crossing to the opposite side of the brain, but they will remain in the same side of the brain when we inject one single eye. Whereas in the case of the albino, we don't see this. In the case of the albino, eventually nearly all of these fibers are crossing to the other side. So we can investigate as well these abnormal chiasmatic pathways. Another feature that we have with mice is that we can genetically manipulate these animal models. Since 1980, and actually this is the front cover of Nature, the journal Nature, displaying one of the most famous transgenic mice. This was a transgenic mouse that was generated with an additional copy of a growth hormone gene. That is why this mouse was 2.5 times bigger than its litter mate that was not transgenic. So this is a rather easy and straightforward micro manipulation in which we can microinject this fertilized oocytes, this one cell embryo, and we can deliver our transgene in one of the nuclei. And this is how we generate transgenic mice. So a transgenic mouse is nothing else but adding another construct, another DNA construct. For instance, we know an albino mouse, and maybe it's due to the mutations in the tyrosinase gene. So we can restitute, we can add a functional copy of tyrosinase, and the resulting transgenic mouse will be pigmented. But we can also play with some extra genes that are not necessarily from the mouse genome. This is a gene from a jellyfish. This is called green fluorescent protein. And we can add this green fluorescent protein gene to a mouse, and we can generate a fluorescent green mouse. And this is something we have done in the past, and this has been, again, instrumental towards understanding of many different diseases and many different pathologies, including albinism. So the question is, how have we been working towards understanding albinism using some of these animal models? Here you have 4 different mice, and there are 3 different types of albinism that have been modeled in this picture, as we will see in the following images. What we can do with transgenic mice, we can play with the level of expression of the transgenes so that we can mimic that, similar to what happens to people for albinism, for oculocutaneous albinism type 1, in which the mutated gene is tyrosinase. What you see is there are different types of expression, different amounts of expression, and we can sort of ask to every one of these mice what is the level of vision that they have. For instance, in some years ago, we were also investigating the role of the regulatory regions, because we were deleting, we were inactivating these regulatory regions, and we were generating these YRT4 mice, the mice in the middle, this mouse in the middle, in which we have a transient or a substantial loss of pigmentation, but it's not yet totally albino. So this would be similar to what happens to the OCA1A-B type of albinism. So this is a person with oculocutaneous albinism type 1 without any a sign of pigmentation. So this is what we normally interpret it as OCA1A. And this is what is being reproduced in this animal model. This is the third mouse counting from the left. Here we have two boys that are having some pigmentation. Nonetheless, they have OCA1 because they have mutations. They have different mutations in the tyrosinase gene. And this additional or this residual enzymatic activity is what is called OCA1B. So we can reproduce this into mice. This is this light gray mouse that is at the border, and this is a good model for OCA1B. Similar, this is albinism in Africa. This is in mutations with OCA2, and we can reproduce, we can generate a mouse that has similar mutations in the OCA2 genes. And I want you to pay attention to the color of the mouse hair and to the color of this person from Cameroon. This is a black person from Cameroon, of course, a black person with albinism. And as you can see, there is a very nice reproduction of the color of the hair. So this is also, we can go beyond with the OCA3, in which the mutated gene is TRP1, and we have the corresponding mouse models. And again, if you compare the color of the hair and the color of these boy hairs, and you will see that they have some similarities. And last but not least, the last example I want to show you is the OCA4, another gene in which we can mutate and we can reproduce the same features that are seen in the human beings. What else we can do with mice? We can do precise mutations, and this is due to the so-called genome editing, and this is now being made possible with the help of the CRISPR-Cas9 tools that were brought together by these two researchers, Emmanuel Charpentier and Jennifer Doudna. And these two researchers were awarded the Nobel Prize in Chemistry in 2020 because of these discoveries. So this is the CRISPR-Cas9 system that we can use in mice to better reproduce different features that we see in albinism, such as trying to evaluate the role of some additional regulatory elements. And this is something we have investigated extensively in my laboratory. And in this case, what we have done is the removal of 5' regulatory DNA sequence. And as you can see, there is an obvious element that has a relevant role in the control of the expression of the tyrosinase gene, because when we remove this regulatory element, what we see is that there is a lack in pigmentation. We can even investigate where this lack of pigmentation occurs. And for instance, if we do histological analysis in the eye, we have the two layers where pigmentation is present. We see the RPE, the retinal pigment epithelium, that remains pigmented. And we see the melanocytes that are colonizing the choroids that in this case are affected. We can investigate in mice these differences in melanin in the skin, and we can quantify these. We can quantify the differences in melanin in the retina in different CRISPR mice in which we are investigating the role of these elements. And we can even do point mutations, such as the so-called avatar CRISPR mice, in which we can reproduce human mutations in these animal models. I'm using this metaphor of this science fiction movie in which this blue beast were associated, each one of them, with a human being. So this is something we have done in different formats and for different types of albinism. This is one of the first mice we produce in the lab, in which we reproduce this OCA4 situation. These OCA4 mutations are deletions of a single cell, a single cytosine. And this is something we have done with several other types of albinism, such as even reproducing these very common variants of the tyrosinase, 192 and 402, that we have eventually been able to determine that these are they need to be considered pathogenic. We can also do some further labeling to investigate these connections between the retina and the brain. And this is again dye-eye anterograde labeling. This is classical anterograde labeling. This is with a Fonda mouse. And we can do in collaboration with Alexandra Repsan and Virginie Chotard from Institut de la Vision in Paris, in France. We can do this anterograde labeling with different colors, injecting different fluorescent colors in both eyes. And so we can illuminate and we can visualize the entire visual tract so that it can be further analyzed. With all these different avatar mice, we can test different drugs and we can test different molecules. Something we have done in the past, we have been investigating the role of the intermediate metabolites in the pathway of melanin biosynthesis. And some 16 years ago, we were the first to realize that it was not melanin, the main culprit of these visual alterations, but one metabolite that was in the beginning of this pathway. And this is L-DOPA. When tyrosinase is not operating, it's not only the disappearance of the end product, melanin, but also the intermediates. Therefore, DOPA is gone. So we try to ask how we will be performing if we would be able to restore the levels of DOPA without affecting melanin, without producing melanin. And we came across an elegant solution, what I think is an elegant solution, which was overexpressing tyrosine hydroxylase, which can accomplish the first step that is normally done by tyrosinase in the retina, oxidation of the amino acid tyrosine to DOPA, but tyrosine hydroxylase, in contrast to tyrosinase, cannot further oxidize DOPA. So we were producing, using albino mice, we were producing transgenic mice with this tyrosine hydroxylase that they had a restored vision, but they didn't produce melanin. So by this very clear experiment, we demonstrated that the most important molecule was DOPA. It was not melanin. In this case, we were able to restore these ipsilateral projections, these chiasmatic projections, and even we did behavioral test and optomotor, so-called optokinetic test, and we can confirm that these transgenic mice, even though they remain albino, because they produce a higher level of DOPA in the retina, they had a better visual system. So this was leading to the establishing of some clinical trials, and this is a clinical trial that was run by Gail Summers and Murray Bridgen, and that was the first clinical trial that was administering L-DOPA to adult people. What happens is that even though they saw some tendency, they saw some trend, and especially with a high dose of DOPA, they couldn't confirm that was a statistical significance difference. So possibly this was due because they were mixing up different types of people with albinism. If they would have done this experiment probably with only people with OCA1, perhaps the result would have been clearer. But this remains to be repeated. Nonetheless, in mice, the team of Elena Lee, they demonstrated a couple of years ago that administering DOPA immediately after birth, and this is up to P15, so 15 days after birth, they would rescue the visual OCA1 phenotype. But if they administer this DOPA later than this, later than P28, so this is about 2 years in humans, they did not see any obvious improvement. So this experiment, it shows that you can play with DOPA and you can add DOPA immediately postnatally, at least in mice, and this has some obvious effects in the restoration of this visual phenotype. This remains to be seen and remains to be tested whether any of these can be reproduced in human beings. Brian Brooks, using also animal models, had a brilliant idea of using nitisinone, which was meant to be tested to be treating tyrosinemia hereditaria type 1 for blocking the degradation of tyrosine. But this drug had another secondary consequence, was raising the levels of tyrosine in the blood. And if there was a remaining enzymatic activity of tyrosine, such as in the case of OCA1V, you will be able to use this excess of tyrosine and produce more pigmentation. They nicely demonstrate this with these Himalayan mice, with mice that represent OCA1B, and they also managed to get some eye pigmentation, even though it wasn't clear, that these mice had, beyond having some increased pigmentation, whether they had a better visual system. We also tested these OCA1B models with nitisinone. We were using the extreme dilution model, which is another OCA1V type. And even though we could demonstrate that we could increase the levels of tyrosine 3 times in plasma after injecting this nitisinone, we failed to see any effects, any obvious effects. So Brian Brooks also was testing this with another type of albinism in mice that were OCA3 models, and they also failed to reproduce. So nitisinone, it remains to be seen whether this has a universal effect with different types of albinism, or it might be only concentrated in some types of albinism. Brian Brooks, they ran a clinical trial on a very limited number of individuals with albinism, and at least in one case they could see some restoration of the pigmentation, as you can see in this progress of these pictures. So it remains to be seen whether nitisinone would be something to consider for a further therapy, not only for restoring pigmentation, but also for restoring the visual capacity. Let me finish by some of the attempts that have been made for treating albinism, or treating albinism with gene therapy. So this is not albinism, this is Leber congenital amaurosis type 10, and this is using CRISPR tools for restoring a given mutation. This is injecting these CRISPR tools intravitreally and subretinally, and this was done by 2 companies, and it was announced at the end of 2018, and this has been developing, and recently, last year, they were reporting some of the first results. And in this case, these CRISPR tools, they were able to remove a cryptic aberrant signal that was present in the gene, in the CFP290 gene that is causing these Leber's congenital amaurosis. And we can think that a similar approach could be used for albinism. In all these cases, the CRISPR tools are delivered through a vehicle, and the vehicle is this adeno-associated virus, AAV. We have different serotypes, and some of the serotypes are targeting the retina cells. So this was exactly what was used like 15 or 16 years before by the team of Enrico Surace in Italy, in Napoli at the time, in Naples, and he was using a mouse model for ocular albinism type 1 and a mouse model for OCA1 with mutations in tyrosinase. And in both cases, they were using AAV, they were using these viral vectors to transduce functional copies of the corresponding mutated gene. So in both cases, they saw a transient improvement in the visual systems of these animals, leading to the conclusion that it could be in the future be possible to do gene therapy using these vectors, perhaps with transgenesis or perhaps with these genome editing tools, CRISPR. Well, beyond using these AAVs, we can also use nowadays these lipid nanoparticles. These are the same vehicles that we have been given, administered as these COVID-19 vaccines. So the ones that are generated by Moderna and by BioNTech Pfizer, they were using this vehicle. This is a non-viral vehicle, and this is probably the future. So we will hear a lot about using these lipid nanoparticles for transducing functional copies of the genes, and also perhaps for albinism. So let me finish by saying that we have learned a lot using animal models on the different features of albinism. We have learned a lot about some preclinical studies towards very elegant and very promising treatments for albinism, but we still are far from reaching a situation in which we can begin clinical trials with safety and efficacy for validating these drugs for human beings. So far, none of the clinical trials that has been run has been successful per se, but what it means is that we need to still investigate using these animal models, and we need to be clear, and we need to know how we can develop and how we can improve these different therapies in order that sooner or later, and better sooner than later, we can eventually test these proposals with human beings in a very controlled way. So let me finish by thanking all my team and the different institutions that are providing us resources for doing all this type of research. And I thank you very much for your attention, and I hope you enjoyed this video presentation on animal models of albinism.